we're seeing degeneration. Like, are you familiar, obviously, with like neutral mutations?
Am I familiar with them? Yes, the vast majority of mutations are neutral. They don't do anything. Although they can come to have function, as in the case of the codfish. It had neutral mutations in this section of DNA, but eventually it came to express this new function. Same with bacteria. You can replace their promoter sequences with like 100 base pair sequences, and within like a few mutations, it becomes a functional promoter sequence.
But aren't we seeing, say, like with the Encode project, that most nucleotides, they're playing now a role, say, in multiple overlapping codes. So that means the beneficial mutations that we were kind of talking about earlier, which are not, say, deleterious at some level, aren't they vanishingly rare? Like, how often do we see it? Because obviously the majority of mutations, like you said, are neutral, and I agree with that. Are the beneficial mutations, the ones that would be amplified, say, by natural selection, are those more so rare?
Are they rare? Yes. Both harmful and beneficial mutations are more rare than neutral mutations, because the code is, because a large portion of our code doesn't do much of anything, and because even the amino acids that are important to us are called redundant or degenerate. Which sounds very malicious in my opinion, but that's one of the words that describe them.
But the thing is, we're seeing
hold on. Well, okay. I want to explain why this is important. Mutations are often neutral, not just because of this large portion that doesn't do anything. They're also neutral. Mutations are neutral because of a redundant amino acids code, because you can have a whole bunch of different sequences that specify the exact same amino acid. Leucine, I think, is specified by 6 different sequences. And so, if you were to change one of the nucleotides in the codon specifying leucine, then you might just change it to another form of leucine, or to another codon for leucine. In which case, you still got leucine. Nothing changed, and so that's why the vast majority of them are neutral.
But the thing is, we're seeing now that a lot of these, say, redundant genes or regions, these, I guess, junk DNA, which would constitute a bunch of different things. We're actually seeing that they play important regulatory roles. We see that they're playing roles in even embryological development, gene expression. So we're actually seeing that the vast majority of our genome is functional to some extent, which would indicate that these neutral mutations are actually more near neutral, which means they are deleterious. We're losing information. So the best way I could put it, the bottom line is that selection, it's going to remove only, say, the worst deleterious mutations and then amplify only the best beneficial mutations. So the best way I could put it is, this means that the accumulating damage is largely invisible, because natural selection, it's not really going to act on these near neutral mutations. They're unselectable, kind of like rust on a car, while adaptations tend to be highly visible, like antibiotic resistance. So even if you, Jackson, say, present me with, I don't know, even 1,000 examples of, say, adaptation via beneficial point mutation, whatever it is, you're still failing to address the key issue, which would technically be net gain versus net loss. Because adaptation, obviously, it explains the fine tuning of the environment, because that's what we're looking at, is fine tuning. But it doesn't really explain the astounding internal workings of life in general. Okay. It does not begin to explain the mystery of the genome.
Okay. Okay. Well, you've covered an incredible range of topics.
I think it just comes down to, is our genome degenerating, right?
Okay, it's not. Genetic entropy is bogus. No geneticists take it seriously except.
Yeah, but I just demonstrated that it actually is, because these near neutral mutations, they're not selected against or for.
Okay, what Sanford does is Sanford ignores beneficial mutations. They're not part of his model because
you just admitted they're rare.
No, I said that they're less common. I mean, in a sense, they're rare. It depends on what you mean as beneficial. But okay, first, for one thing, you're bringing up a lot of topics and bringing them up in sort of ways that, for instance, let's go back to Encode for a moment there, because you brought that up. Do you know what Encode meant by functionality? When they said something, a section of DNA was functional, do you know what they meant by that?
Well, I know that they're playing more important roles, say as regulatory roles, gene expression roles. These are still roles in general. I know what you're going to get at. You can explain it, but go ahead.
So when they said something was functional, they meant it was transcripted, which is indeed a large part of our gene, or a large part of our DNA is transcripted.
Yeah.
But that doesn't, parts that don't do anything are transcripted. I
just told you they do stuff. They play important regulatory roles. We see that they're playing roles.
No.
Embryo. This is function, Jackson.
No, not actually. A lot of the parts of our DNA don't have any function. That's why the pseudogenes, for instance, don't have function. ALU retrotransposons don't have function. There are lots of retrotranspositional parts of our genome that don't have any function.
These leftovers that you're talking about, they do have very important functions.
Actually, a lot of them don't, but that's not the point. The point I'm trying to get is, and I can demonstrate a lot of them don't have function. For one thing, actually, I want to finish first because this encode thing is really important. They defined functionality in such a way that it's virtually useless. Is it technically true? Sure, you can define functionality as it's transcripted and then say a large part is functional. That's not technically wrong. It's true. A large part is transcripted. But so what? That doesn't tell us literally anything about about differentiating the parts that do work from the parts that don't work. For instance, we have the gulo pseudogene, which allows us, or which doesn't allow us to synthesize ascorbic acid. I mean, the parts of that is still transcripted, but it's still functional according to Encode, but it doesn't do anything. It's literally broken. It has no function. And yet it has no function in the sense that it's not doing anything, and yet they call it functional. Would you consider that useful?
Well, let's just say, because according to my model...
Yes or no, would you consider that useful?
The thing is, we only speak 1 to 2% of the actual, DNA
yes
language.
Or no, would you consider that useful?
It could have been at one point, and now we're experiencing the degeneration. Let me just say,
when Encode did their project, where they declared that such segments as gulo were functional, would you consider that useful?
It depends what the use is, because like I said,
it has no usage.
1 to 2% of the DNA.
It's very well studied and very well understood. It has no function. Okay, so yes or no, is it useful to describe it as functional?
For argument's sake, let's say that pseudogene, that specific one, doesn't have any function. Let's say it, but all I'm saying is, my Honda Civic, when it came off the assembly line, it worked great. Air conditioning was working, brakes were perfect, no rust. Over time, the car has degenerated. The air conditioner broke. That just shows evidence of obviously degeneration from the fall that we have experienced these accumulations. So if something's broken, it doesn't really provide evidence for that type of evolution that you're talking about.
I'm not talking about evolution. I'm talking about is it useful to say a thing that has no function is functional? Is that a useful construct?
I'm saying that I'm going to expect
yes or no.
If there's no actual, say, regulatory role, some type of role in the embryo,
it doesn't make any proteins, it doesn't regulate any other genes, it does nothing.
I'm not saying that our genome is 100% functional, but I'm saying enough of it is functional where those neutral mutations are now near neutral mutations. They're building up in the population. We are degenerating. I mean, are you denying that?
Am I denying what? That neutral mutations are occurring in our population? Absolutely not.
But population geneticists themselves even agree that we are degenerating. I mean, we are accumulating.
Which population geneticist? John Sanford?
John Sanford references many population geneticists that are actually evolutionists in his book. They're not even creationists.
Kimura, but he misunderstands and misrepresents their models to make his argument. For instance, he ignores beneficial mutations.
He already admitted they're rare.
They're not just rare, they occur.
But the thing is, natural selection will amplify those, but those millions of deleterious near-neutral mutations. They're building up. You can't select those out.
That's not no
possible ways.
What the point is with beneficial mutations is these things are. I will ask him, RJ. The beneficial mutations outweigh a lot of other mutations, and that's part of what researchers have discovered. Have you actually gone through the sources that Sanford puts forth in his arguments and read what these other population geneticists like Moto Kimura have said?
I've read what they said in, I know there's a rebuttal to John Sanford's book. I think the guy's name was, I don't know, Scott or something, some type of blog. So he had some references, but no, I've never gone and actually like read their papers or anything.
I would really recommend it. Go to Google Scholar, actually read the papers that he is referencing, and go see for yourself if what he's saying is true. I always go to Google Scholar when I see something that someone has written about a paper. Like even when I get because I follow Science Magazine, even when they send me like their secondary sources, like somebody wrote an article about a technical paper, I don't want to read that article they wrote. I want to go and read the technical paper so I can see if they're accurately presenting it. You see what I'm saying?
No, I understand that, right. But I mean, and break it down for me, because obviously, you know your stuff here. This is what your education is. Well, but if I'm making it simple, like, and just show me where I'm wrong here. I just kind of want to make an analogy. So okay, go ahead. I'll go after you're done.
The major problem with Sanford's argument, and I'm not saying beneficial mutations aren't rare. In a lot of cases they are. I mean, we're a lot less likely to hear about beneficial mutations occurring in human populations, even though they do happen, but they don't happen very often.
I just want to correct one thing. I'll let you continue. When I say beneficial mutations, I more so mean like a beneficial outcome, because a lot of times these beneficial mutations are due to a loss of function and a loss of information. So you're still not getting rich by losing money.
I mean, although in a lot of other cases, they're caused by, for instance, gene duplications where you're adding segments and whatnot. For instance, the apolipoprotein A1 mutation in a family in Italy. Have you heard of that one?
No. You want to explain it?
Okay. So for anyone who doesn't know, the mutation, we all have a gene that specifies this protein called apolipoprotein A1. Basically, what it does is it allows us to digest cholesterol. So there's a family in Italy, and they had a mutation in their apolipoprotein A1 gene. It switched out an arginine for a cysteine. Basically, it was just this one little point mutation, and it allows they are now more likely, they're better able to digest cholesterol, to digest larger amounts of cholesterol than people are generally, which means they are less likely to get heart disease, for instance.
But it still sounds like I don't know that exact one, but it still sounds like if I look at the aerobic citrate digestion, that involved the loss of the control of the normal anaerobic citrate digestion. But what you're saying, it just seems like if I look in the literature, and I will, I mean, if it's a loss of control of something, if it's a gene being turned on and off, it's not really novel.
They didn't lose control of the gene, though. The gene's function was amplified.
Exactly. So it's amplified and emphasized based on, say, whether it's decompressing or unscrambling of existing information. That's not going to get your bacteria to biologists. But that's just still consistent with my model. Go ahead.
Okay, but remember, I gave an example earlier with the notothenoid fish of a section of nucleotides that did nothing, that became a functional gene. And this happens a lot. It happens in bacteria. There's actually a really interesting paper published only a few months ago about inserting 100 base pair sequences instead of promoters in bacteria, and within like 1 or 2 mutations, they turn that sequence that is random nonsense into a promoter sequence. They're not doing it consciously, of course. They have no brain, they have no control over their own genetics, but mutations are occurring such that these sequences, these segments that don't do anything, have become functional. And that's really not all that uncommon. I mean, sure, it's rare, but it does happen. notothenoid fish is just one example. It's a really interesting example. But we do see examples of new structures, new functions, sequences that had no function that become functional.
Are these the types of changes that are going to build the genome, though, that you're
what do you
telling
mean by build the genome?
Well, obviously, we're going to have to explain, this information system chock full of even meta information, overlapping codes. I mean, if we look at the phenotype, are these changes producing any type of significant phenotypic change, or is it just
sometimes? For example, in I think Midas cichlids, they have a lot of phenotypic plasticity in their jaw shape, which has contributed to how they speciate. And so, yeah, in some cases, yeah. Well, in another case, the lizards, the Italian wall lizards on Podmakaru are another example where the scientists took a very small population of these lizards from this nearby island Podkopiste, and it's like 5 males and 5 females, and they dropped them off on Podmakaru in the 1970s, and in just over 30 years or so, these guys developed novel structures, and they directionally they underwent directional selection towards becoming herbivores, and they even developed a cecal valve, which is what herbivores use to digest their plant material, which because the original population was insectivores, so they underwent directional selection and developed new structures to accommodate that.
Well, on that one, like, if I could just comment on the lizard one, because I think that's a common one too. Like that, let's say new or novel, let's say muscular valve that they found between, I think it was the small and large intestine. If you actually look at that, it's really just an enlargement of muscles already present in its gut wall. So in other words, far from being, a truly new novel feature like you're saying, the shift in available food, Jackson, allowed the lizards with larger muscles at this juncture I'm referring to be more successful at feeding and reproducing. But the important thing is, let me just finish.
Okay,
if these lizards were returned to their original habitat, that cecal valve feature, which is cool, it's a cool variation based on, it's not proving any type of Ponce come to people evolution, but that may actually dwindle based on the lizard's return to an insectivore diet, like you just said. So, I mean, that's not really,
we don't know that though.
Change.
That's an assumption, but we don't have any evidence to indicate that would actually be the case. But again, I'm left to ask, what would you expect as an example of something that in your mind would allow Ponce to people evolution? Because I've given you examples of new structures that have arisen through mutations. I've given you examples of non-functional sequences that have become functional. So what sort of mutation are you expecting?
Well, I'll give you an answer to that. I just want to, because we kind of went on, well, I mean, we've been on a little bit of a few rabbit trails. I just want to say one last thing about the genetic entropy, because when it comes to the near neutral mutations and when it comes to the beneficial mutations, like you're saying that he ignored, obviously a species, Jackson, it can undergo the minor adaptive fine tuning to its environment, like you're saying. I mean, that's observable. I agree. But at the same time, okay, the degeneration process is going on in many other ways. Like, to make it simple for the audience, because some of this I'm sure would go over people's head. Like, picture a 10-year-old car. It is degenerating in all possible ways, Jackson. Whether I install new windshield wipers, the question is, has the car stopped degenerating? I'm going to ask you that question. Has the car stopped degenerating if I change the brakes or switch the tires out?
You're going back to analogies. We need to look at the actual data. Analogies are fine up to a point. I don't really like to use analogies when I'm talking to people, when I'm explaining to people scientific concepts, but I think it confuses the issue. I really honestly do think it confuses the issues.